So far in today's class, you've been looking at the two basic cell types, which are prokaryotic cells and eukaryotic cells, and hopefully you have a pretty good understanding of the basic differences between those two cell types. At this point, we're going to shift gears to looking just at eukaryotic cells, and we're going to be comparing plant and animal cells. When you come to class next time, you are going to be doing a lab called Comparing Plant and Animal Cells, and you're going to be learning about the structure of those cells, and you're also going to be working on proper drawing and labeling of those cells, which are both important IB biology skills. As a part of that lab, you're going to be looking at these 6 different cells under the microscope. Ahead of that, you need to predict which organelles you might see there, and then you're actually going to go look for those organelles under the microscope, as opposed to me telling you what's there ahead of time. So to prepare for that, I'm asking you to do some note-taking, and you have a note sheet. This is a portion of it that you should be filling in today to get ready for the different types of cells that you'll be looking at. This note sheet asks you to do some drawings in some places. It asks you to write out some functions, and the really important part for our lab next time is this last column where you're identifying whether the organelle is found in plants, animals, or both. You obviously can use the internet and Google things, but I'd also like to encourage you to use the online course companion, which you should have access to, as well as another textbook that we have in class called the Pearson textbook. You do not have online access to that, but I have put the relevant pages on Canvas for you for topic 1.2 Pearson e-text, it's called. Here's a picture from that Pearson text of an animal cell, and you can see it has the organelles nicely labeled, and in some cases it has annotations which describe either the structure or the function of that organelle. Here is the same picture for a plant cell, and again you can see labels and some annotations. Your goal is to understand not just the structure but the function of each of these things, and so in some cases these annotations would give you all the information you need, and in other cases they wouldn't. When you are presented with an organelle that doesn't have a function with it, then you'll need to look at other areas within the textbook or other areas online to find those functions. So I'm going to model for you how I would go about figuring out the functions for endoplasmic reticulum. If you look up here, you see that it describes that it's a network of tubes and flattened sacs, and it says that it may be smooth or it may be rough ER, but it doesn't really tell us exactly what it does. And so to answer the question of function, I'm going to go take a look at a different part in the Pearson textbook, and I will model for you how I would go about determining the functions. So I have turned to the Pearson text to page 18, where I see the picture of the ER, and I also see some description here. And as I read through this, the first part is about smooth ER, and it talks about the production of membrane phospholipids and cellular lipids, and I know lipids are fats. It talks about the production of sex hormones, detoxification of drugs in the liver, storage of calcium ions in muscle cells, and so on. As I'm reading this, I see that some of these bulleted points are highly specific. For example, testosterone and estrogen are only produced in the testes and the ovaries. They would not be happening at smooth ER in cells in other places of my body. And I can see several of the others are specific to the liver or the muscles. So as I'm taking a look at this, I want to focus in on general functions that would be true for the ER in pretty much every cell of a eukaryotic organism. And so as I take a look at this, I see here smooth ER, I see the production of membrane phospholipids and cellular lipids, and I see transportation of lipid-based compounds being two things that are fairly specific to ER are fairly general for ER in any cells of our body. Next, I'm going to read about the rough ER, and again, it talks about the fact that it has ribosomes on the outside of it, and we know ribosomes are involved in protein synthesis, so it says, therefore, this type of ER is involved in protein development and transport. So I'm thinking that that's probably the most important things to remember about the rough ER, that they're involved in protein development and transport. So now that I've done my reading and highlighted some important things, I'm going to come back over to my note sheet, and I'm going to type in a summary of what I've just read. So rough ER first, I'm going to say protein manufacturing and transport. For smooth ER, I'm going to talk about producing lipids for the cell and membrane and transporting them. So those seem to be nice, simple summaries of what the rough endoplasmic reticulum and the smooth endoplasmic reticulum does within most generalized eukaryotic cells. So you will want to continue in that way for all of the organelles in eukaryotic cells and be thinking specifically about whether they are in plants, animals, or both. And the pictures that I showed you from the Pearson book would be very helpful to you for that also. And you'll note that this says that this is a level 1 assignment. That means that you may write your answers straight from an internet source or straight from a textbook source. You don't necessarily have to put them in your own words. However, you should not simply copy somebody else's paper, as that is not fair to the person who did all of the work. So you don't have to paraphrase, you don't have to summarize necessarily, but you still should obtain the information on your own. If you haven't done so already, make sure that you and your partner have worked out the corrections for your functions of life labs, and make sure that you finish your cell type versus cell size labs so that I can take a look at those and put some comments on there for you. Please come to class with this note sheet pretty well underway so that you have it as a reference for the lab that we'll be doing.